In this video also we will discuss about one of the inhibition strategy that is used to prevent SARS-CoV-2 virus to get into the cell. And for this, what researchers have done is that they have tested various interferon mediated gene. So what is interferon? Interferons are generally expressed by the cells on its own whenever they encounter a virus entry. So what researchers have done in order to identify which interferon is much more impactful or is helpful in inhibition of SARS-CoV-2 virus to get into the cell, they have artificially tested hundreds of interferons, and they have find out that lymphocyte lymphocyte 6 from a family of E, lymphocyte 6 family E, or in short this helps in inhibition of SARS-CoV-2 virus inside the cell. And the spike protein of SARS-CoV-2 virus, one of the characteristic of spike protein of SARS-CoV-2 virus is that it is heavily glycosylated. Problem because of this heavily glycosylated is that there are several epitopes even though if some epitopes are available that are recognized by immune system of the body, that will initiate the immune response in the body. However, because of this heavy glycosylation, the epitopes are not being able to get in touch with immune cells, or we can say that immune detectors that like B lymphocytes or T cells that detects whether we have a foreign particles inside the cell. In order to detect that part, they need to recognize the epitopes of the foreign particles. And in this case, SARS-CoV-2 virus, because of heavy glycosylation, that epitopes does not or there are very less chances that epitopes comes in contact with the immune system of the cell. So, and same is the case happens with HIV. In HIV also, our body is not able to detect that it is that something foreign particles has entered inside the system because of the heavy glycosylation. What that it means that suppose this is the chain of amino acid, and we have understood that N glycosylation occurs at NXT site, and O glycosylation also occurs in Golgi apparatus. And if this is heavily glycosylated, the immune system or the T cells or B cells will not be able to come in contact with these epitopes. Suppose this is the epitope, so there are very less chances that this immune cells or immune sensor will be able to recognize the epitope. So because of this, most of the time SARS-CoV-2 virus gets undetected at the initial stage of the infection. And this is also the reason that symptoms start occurring very late, or we observe delayed symptoms, because our immune system will recognize this virus when the infection rate has or the SARS-CoV-2 virus has infected an individual to a substantial extent. And before SARS-CoV-2 virus, there is a SARS-CoV-1 virus also there. SARS-CoV, okay, so that is only called SARS-CoV virus. And for SARS-CoV-2 virus, people have been able to make a monoclonal antibodies. That can specifically target the some of the epitopes of SARS-CoV virus. So when people have tested these monoclonal antibodies with SARS-CoV-2 virus, they find that these monoclonal antibodies is also helpful in reducing the infection of SARS-CoV virus. And so now what people are using, people are trying to use different antibodies at the same time. Not only this, and other antibodies. In such a way that they are while designing the experiment, they are ensuring that whatever the antibodies they are using, they are not competing for the same epitopes. Consideration. Target different epitopes. So by this they can increase the inhibition rate or inhibition efficiency of SARS-CoV-2 virus. And one more thing, another strategy that people are using is that the sera or the serum that people get from that one can get from the people who are earlier affected by SARS-CoV-2 virus has been proved to be successful in preventing the SARS-CoV-2 virus in another individual. So sera from already infected person can be used to for another individual to prevent them from SARS-CoV-2 infection. What is sera? Sera is actually whenever someone gets if there is a blood and we centrifuge it, then the blood will coagulate at the bottom because of the density, and we have some supernatant, and supernatant is called as sera. Suppose this is the tube, and after centrifugation we will have blood that is separated at the bottom and have some supernatant that is called as sera. Supernatant. Supernatant that is called as sera. So that can be used from an already infected person to prevent another individual from getting infected with SARS-CoV-2. That's all for this video. Thank you.